Welcome to this lesson on PCR, which stands for Polymerase Chain Reaction. This is one of the most revolutionary techniques in molecular biology that allows us to amplify DNA segments in the laboratory. By the end of this video, you will understand how this powerful technique works and why it has become so essential in modern biotechnology. PCR is a molecular biology technique that rapidly amplifies specific DNA sequences through repeated cycles of heating and cooling. Think of it as a photocopier for DNA. What makes it truly remarkable is its ability to create millions of copies from a single DNA template in just a few hours. This means scientists can take a tiny amount of DNA and produce enough copies to study and analyze. To perform PCR, we need 4 essential components. First, we need template DNA, which is the original DNA sequence we want to amplify. Second, we need primers, which are short DNA sequences that flank the target region and tell the system where to start and stop copying. Third, we need DNA polymerase, which is the enzyme that actually synthesizes new DNA strands. And finally, we need RT-PCR, which are the building blocks, the nucleotides, that are used to construct the new DNA strands. PCR works through 3 repeated steps in each cycle. Step 1 is denaturation. Here, we heat the reaction to 94 to 96 degrees Celsius to separate the double-stranded DNA into 2 single strands. Step 2 is annealing. We cool the temperature down to 50 to 65 degrees Celsius to allow the primers to bind to their complementary sequences on the DNA. Step 3 is extension. We raise the temperature to 72 degrees Celsius, which is the optimal temperature for DNA polymerase to synthesize new DNA strands. Each cycle doubles the amount of DNA. The key to PCR automation is an enzyme called Taq Polymerase. This is a heat-stable enzyme that was isolated from Thermus aquaticus, a bacterium that lives in hot springs. What makes Taq polymerase so special is that it can survive repeated heating to 94 to 96 degrees Celsius without denaturing or breaking down. Before the discovery of Taq polymerase, scientists had to manually add new enzyme after each cycle, but now the process can be fully automated. PCR amplifies DNA exponentially, following the formula 2 to the power of n, where n is the number of cycles. After 20 cycles, you get approximately 1 million copies. After 30 cycles, you have around 1 billion copies. And after 40 cycles, you reach approximately 1 trillion copies. This exponential growth is what makes PCR so powerful, allowing us to work with even the smallest DNA samples. PCR has numerous applications across different fields. In medical diagnostics, it is used for detection of genetic diseases and pathogens, including viruses and bacteria. In research, scientists use it for gene cloning and sequencing studies. Forensic science relies on PCR for DNA, fingerprinting and identification of individuals. In agriculture, it helps with GMO detection and plant breeding programs. PCR has truly transformed how we work with DNA. Over time, scientists have developed several variants of PCR for specific purposes. Real-time PCR, also called qPCR, monitors the amplification process in real time, allowing for quantification of the original DNA amount. Reverse transcription PCR, or RT-PCR, first converts RNA to DNA before amplification, which is useful for studying gene expression. Multiplex PCR can amplify multiple target sequences simultaneously in a single reaction, saving time and resources. PCR offers several key advantages. First, it is incredibly fast, providing results in hours rather than days or weeks. Second, it has high specificity, meaning it targets exact DNA sequences with great precision. Third, it is extremely sensitive and can work with minimal DNA samples, even from a single cell. Finally, the technique is fully automatable using thermal cyclers, which are machines that control the temperature changes automatically, making the process reliable and reproducible. PCR is truly a revolutionary tool in molecular biology. It is a simple yet powerful technique that has transformed research, medicine, and forensics by enabling rapid DNA amplification from minimal samples. Since its development, PCR has become an indispensable tool in laboratories worldwide, contributing to countless discoveries and applications. Understanding PCR is essential for anyone studying biotechnology or molecular biology.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.